All right, so you should have, hopefully, your chapter reviews out in front of you. We will go over the answers. All right, so the first one, the chromosome theory of inheritance, is that genes are carried from parent to offspring on chromosomes. That is a summary of what it states. Mrs. Miss. Yes. Okay. Yep. Bye. We have a meeting over by the library. So it's in like that little hallway if you go to the library. Okay. Meiosis then is defined as the process by which the number of chromosomes is reduced by half to form sperm and egg cells. Okay, hopefully you have something similar to that. I'm going to make our text a different color. All right. How many pairs of chromosomes do human body cells contain? You're looking at the pairs. 23 pairs, okay? Oops. Struggling today. All right. Make sure you understand if it says pairs or like individual, you know the difference. Where do chromosomes in each pair come from? Where do you get your chromosomes from? Allie? Your parents. So one, I guess we could say parents. You get one chromosome from the male parent and one from the female parent. And then how many genes do our body cells contain? This number varies a little bit. That's a big number. Allie? 20,000. Yep. We're gonna say like 20 to 30,000 plus. Okay, there's a lot. Then, moving on to our WebQuest section. What is it? What is a diploid cell, or what does it mean to be diploid? How would you explain that? Allie, go ahead. Yep, contains 2 copies of each chromosome. So this also means it has a complete amount of DNA per cell. So it has the full amount. Your body cells would be considered diploid cells because they have the full amount of DNA. Then how about a haploid? Haploid cells. Age? Yep. Okay, a cell that only has one set of chromosomes. So that's half the total number or total amount. Your reproductive cells, so like sperm cells and egg cells, are haploid cells because you get half of it from mom, half of it from dad. When it combines, it creates that full organism. Oh, okay. Pull down. All right. The process of genetic recombination. What do we got? Allie? Yep. Okay. When the genetic information is exchanged maternally and whoops, paternally. Okay, so between like mother and father, or from the mother and from the father. Then, true or false, a cell that is not dividing is said to be in interphase. Ansley, that is true. All right. Going through the steps here, I'm going to summarize for you. Okay. In prophase 1, this is where chromosomes condense. They line up in homologous pairs. Metaphase 1, the homologous pairs move to the center of the cell. Anaphase 1, the homologous chromosomes separate, so now you have each like individual side. Each moves to opposite sides of the cell. They're pulled by the spindle fibers. Then telophase 1, the movement of chromosomes is complete. Cytokinesis happens, and 2 cells are formed. So we have 2 cells at the end of telophase 1, and that's cytokinesis. Allie? These ones, I think I would have to check. Let me check here. Yes, they are haploid cells. Okay. Then moving into prophase 2, it's pretty much the same as before. Chromosomes condense again. This time, they're chromatids. Which are like each individual side of the chromosomes. Then metaphase 1 or metaphase 2, the chromatids align in the center and spindle fibers attach to the centromeres, which are what like hold them in the center. Anaphase 2, the centromeres divide, so they're splitting the chromatids. Sister chromatids move to opposite sides of the cell. And then telophase 2, you again have the movement is complete. This time, cytokinesis occurs and 4 haploid cells are created. Okay. Spermatogenesis, that final cytokinesis stage, what is created or what happens? Ansley? Yep. Okay, so it's the development of sperm cells. Specifically, there are 4 individual sperm cells created. Okay. Then oogenesis, what is created? Gage? Yep. The development of one ovum, which is the egg cell, and 3 polar bodies. So they're cells that are created and essentially just dissolve and disintegrate and are absorbed. This also just kind of shows how the egg cell is going to be larger than the sperm cell as well, since the egg cell contains everything that you need essentially for life. Okay, moving on to section 4.4. The summary of 4 steps of protein synthesis. If you guys just use your 4.4 guided reading notes, they just summarize those steps really quick are going to be DNA creates messenger RNA or synthesizes it. Then the messenger RNA attaches to ribosomes. Then transfer RNA attaches to messenger RNA and reads the code. Then amino acids are added to create growing protein. Okay? What are the 4 nitrogen bases that make up DNA? Ansley? Thymine, adenine, guanine, and cytosine. Okay. And then the 4 bases that make up RNA are what? Allie? Yep. Adenine, uracil, guanine, and cytosine. So the uracil and thymine are what change. Then the genetic code is considered a code of bases, so that adenine, thymine, guanine, cytosine, uracil, guanine, cytosine, adenine, that specifies what type of protein is produced. So it reads those nucleic bases, and it determines what amino acid you need. Then, true or false, a group of 4 nitrogen bases codes for 1 amino acid. Allie? Yep, it is false. It should be one group of 3 nitrogen bases codes for 1 amino acid. Right? Then, what does messenger RNA do? What is the purpose of messenger RNA? Allie? No. Not quite. Because it already is RNA. Gage? Yes. Okay. So it copies the coded message from DNA in the nucleus, and it carries the message to the ribosome. Okay, then transfer RNA, what does that do? Go ahead, Allie. It does attach to the messenger RNA. What else does transfer RNA carry? Go ahead, Gage. Yep. Okay, so it attaches to the mRNA and reads the code, then attaches amino acids together to create protein. Then what is a mutation? Go ahead, Ainsley. Any change in a gene or chromosome. Then true or false, whether a mutation is harmful or helpful depends partly on the organism's size. Gage? It is false. What does it depend on? Ainsley? No. Allie? The organism's environment. It depends on the organism's environment. So what it's surrounded by. An example would be if you had an albino or white, let's say like lemur or a koala bear, if that's a mutation causing it to be white, if that koala bear is at the zoo, that white coloring is not going to affect it because the zoo is a safe environment for it to live in. But if that koala bear was out in the wild and it was white, it would be a harmful mutation because it now is something that can like expose the koala bear more. It's more easily seen. It can't blend in, and so now that affects its survival rate. Okay, it depends on the environment. All right. Moving into our protein synthesis worksheet, transcription. What is that process, or how is it defined? Ainsley. Good. So when genetic information is synthesized into complementary RNA, then what is translation? What happens in translation? Stage. Right. So translation is where the messenger RNA is read and it specifies the sequence of amino acids I'm just going to summarize a little bit more there, to create a specific type of protein. Okay? Then, what is a codon? And through that definition, what type of RNA is a codon attached to? Allie? Yes. Right? It is a triplet of nucleotides on the messenger RNA chain that codes for a specific amino acid. Then, what is an anticodon and what type of RNA? If a codon is on messenger RNA, what would the anticodon be? Allie? It is on transfer RNA. So the anticodon is just the triplet of bases or those nucleotides on transfer RNA that recognizes and matches to the bases on the mRNA. So it reads that code, and then it knows which amino acid to add. Okay? All right. This was a warm-up. So, what location in the cell does the first step of protein synthesis take place? Ansley. The nucleus. Which location does the second step of protein synthesis take place? Tony. Cytoplasm. Where in the cell is messenger RNA read and amino acids added? Allie. Ribosomes. Right. Getting into mutations. A substitution mutation. I'm going to go through these quickly. A substitution mutation is a mutation that exchanges one base for another. So it switches them out. An insertion mutation is when an extra base is inserted into a new place in the DNA. So you are adding more bases in. Okay. A deletion mutation then is just like it sounds. When a section of DNA is lost or deleted. Then a frame shift mutation is when deletion or the insertion alters the gene so that the message is no longer correctly parsed, which essentially means like synthesized so it can be read and understood. Okay. And then where can mutations occur? What type of cells can mutations occur on? Allie? Reproductive cells? and body cells. Okay, so a mutation can occur on all different types of cells. If it occurs on a body cell, it's not transferred to the offspring. If it occurs on a reproductive cell, it can be transferred from parent to offspring. All right. You guys then should have gone through the protein synthesis practice using those worksheets and examples. I'm going to just run through the amino acids quick because if you have the correct bases, you'll get the correct amino acids. You should have MET, HIS, ASN, GLN, ILE, LEU, and then the stop codon for the first one. Your bases would be ILE. Okay, so that first one would look like this. I'm going to keep going through here. Your second amino acid should be the MET, PRO, LYS, SER, VAL, PHE, and then your next stop codon. Again, your letters and you'll notice there are different variations of the stop amino acid. Then the third one, your last amino acids, MET, SER, BYS, SER, VAL, PRO, and then your stop codon and your letters. All right.